Hello everybody. Today my presentation will be focused on lipid biosynthesis. This will be the first of the two presentations I will dedicate on lipid biosynthesis. Now before we begin the topic of lipid biosynthesis, I always like to ask these general questions. For example, normally our perception about fats is that we usually connect them with something negative in terms of human health. And that's why I'm asking, are the lipids the root of all evil for human health? And I guess to answer that question, or at least something to help us answer that question, would be to ask another question, and that is, what role do lipids play or have in our body? And you can see here general areas that lipids are involved in. Energy storage, triglycerides. A lot of people, especially in the modern world, don't really worry about that, but this is something substantial. Energy storage is a way to survive, or was a way to survive in the olden days, where food was not easily obtained, and having energy deposits in the form of fat was something that was necessary for life to survive. I mean, if you just think about how much time people spend now versus, let's say, 500 years ago, 1,000 years ago, in terms of obtaining food and spending on thinking about producing food and gathering food and preparing food and what type of food people will eat, you will realize that that time has decreased dramatically. In those days, not long ago, survival was, or having enough energy stored in our body was a matter of survival. Lipids are also involved in the structure of any cell or any body. These are membrane lipids. I mean, think about it. Cells do not exist without cell membranes, which are made of lipids. Hormones, steroid hormones, and other hormone-like molecules are of critical importance for the normal function of a developed multicellular organism. Messenger molecules, phosphatidylinositol derivatives like PAP2, IP3, if you remember from 481, critical role. Eicosanoids, critical role. Vitamins and cofactors, vitamin K, vitamin D. Anchoring molecules, they're involved often in the anchoring of signaling proteins to the membrane, if you remember from 481. Transporters, detergents such as biosalts, critical role for normal digestion. Someone might say, well, I mean, what if the gallbladder is taken out? Well, gallbladder is just a container that stores extra bile that is released when it's needed. When the gallbladder is taken out, actually, the body depends only on the bile that is produced by the liver in small quantities. That's why usually people with removed gallbladder, their diet has to change. They cannot eat as much fat. They cannot eat as much, well, mostly fat, and without meat, because there is not enough bile and bile salts released to help the body to process the fat that is being consumed with food. So when the gallbladder is removed, you're not now bile free. It's produced and still it's released, but in very small quantities. Also pigments, retinol, keratin. Someone might say, well, I mean, pigments, not a big deal, but retinol is actually a precursor for vitamin A, or, well, for the molecule that helps us see. Keratin as well, very important molecule that is used for generating other bioactive molecules in the body. So as you can see, fats are essential for life. There is a reason why fat is the best, fat is the best way to store energy, and that's why our bodies are made to store it. Now, the fact that we as humans abuse food and as a result gain too much storage, if I can use that expression, that's a different story. It has nothing to do with the molecule or the types of molecules fats are. They're essential molecules for the body. As a matter of fact, in 481, if you remember, I introduced to you 2 essential fatty acids. We've never talked about essential carbohydrates because we can make all the carbohydrates we need in the body, and as long as we consume some form of carbohydrates, we'll be fine. Any carbohydrates, except, of course, cellulose or chitin or something like that obviously we cannot process, but anything that is made out of glucose and it has alpha 1 to 4 bond, glycosidic bond, we can make it. We can make any other form of carbohydrate that we need, but that's not how it is with fats. There are certain fatty acids that are critical, and we have to have them in order for us to make bioactive molecules that have hormonal-like properties. So I will let you answer this question: are the lipids the root of all evil for human health yourself? Perhaps for some this is more close to home, to heart. For others that might not be a problem. But it's not only a matter of weight, it's also about a matter of healthy lifestyle and also some genetic predispositions. For example, I've had a friend who is actually quite skinny, but he has a familial predisposition for high cholesterol, and so he was having a very high cholesterol in his blood, even though he was not at all fat. Since high cholesterol is usually associated with overweight, that wasn't the case for him. So there are certain issues that can rise from either misuse of food or certain food groups, or just simply because our body has certain abnormalities. All right, so what are we going to talk about today? We will talk about fatty acid synthesis, and next time we will talk about triglycerides and membrane lipid biosynthesis and cholesterol biosynthesis. So today we're mostly going to focus on fatty acids biosynthesis. Now, to begin that process, it is noteworthy to mention that biosynthesis of fatty acids, it's not going to be the reverse process of beta oxidation. Gluconeogenesis and glycolysis, with the exception of 3 of the 10 reactions, it was pretty much the same. Usually most of the enzymes are shared between glycolysis and gluconeogenesis, but that's not at all the case in fatty acids biosynthesis. First, biosynthesis occurs at a different location. Biosynthesis occurs in the cytosol, and beta oxidation occurs in the mitochondria. Also, a different set of enzymes are used for both processes. A little later, I will make a correlation again with beta oxidation and biosynthesis, which will actually highlight that in principle, they're exactly opposite in terms of what actually happens with the molecule or the molecules that are involved in this process. But in terms of the machinery, they're completely different processes. Biosynthesis in the cytosol, and it's done actually by one enzyme with multiple functions. So biosynthesis, in order to begin, it uses malonyl-CoA. And we've talked about malonyl-CoA before when we discussed the hormonal regulation of the carnitine shuttle. And we said at that time that malonyl-CoA is produced from acetyl-CoA and carbon dioxide. And naturally, I would ask, what do you think the name of the enzyme would be? And since you see the whole slide, that question kind of defeats its purpose because the answer is right underneath. Acetyl-CoA carboxylase. I mean, how easy it is to remember. Acetyl-CoA carboxylase, acetyl-CoA. So for fatty acid synthesis to begin, malonyl-CoA must be generated, because that is the continuous molecule that needs to be fed into the pathway in order to make in order for the fatty acids to continue to grow in size. So how is malonyl-CoA synthesized? This is the structure of malonyl-CoA. You can see acetyl-CoA, basically this portion, and another carboxyl group added, so malonyl-CoA. So to generate malonyl-CoA, we need the involvement of carbon dioxide and ATP. So, and biotin is involved in this process. So what is the role then of biotin and ATP? How do they work? So in this process, ATP would be used to phosphorylate or adenylate, for that matter, biotin. Now, how can we tell what was ATP used for? Well, we look at the product. If the product is ADP and PI, that means that ATP was used for phosphorylation. If the product is AMP and pyrophosphate, or 2 PIs, then that means that ATP was used for adenylation, where AMP is added to the molecule. So ATP was used to phosphorylate biotin. Then a bicarbonate or carbon dioxide ion comes in. Well, carbon dioxide or bicarbonate ion comes in. Someone might say, which one? Well, the most likely form of carbon dioxide in solution will be the bicarbonate ion. I mean, it's a lot higher concentration. As you know, carbon dioxide is very poorly solubilized in aqueous solutions, so it usually will not be carbon dioxide. It will be the bicarbonate ion. So the bicarbonate ion comes in, and then it's added by replacing the PI that was added to the biotin. So we have carboxylated biotin, which changes its orientation because of that. And later on, the process of addition of the carboxyl group to acetyl-CoA is finished, and then we end up with malonyl-CoA. The enzyme again that furnishes this reaction or catalyzes this reaction is acetyl-CoA carboxylase. This is the carboxylase site, and this is the transcarboxylase site. Those are details. I don't want you to worry too much about it. The most important part here is to understand that ATP was used to activate biotin by addition of a phosphate. Then a carbon dioxide will be added, replacing the inorganic phosphate, and then the carbon dioxide will then, or the carboxyl group will then be added to acetyl-CoA, forming malonyl-CoA, and the enzyme goes back to its initial state, ready to accept another molecule of acetyl-CoA and bicarbonate ion and ATP, so that it will form malonyl-CoA. Alright, so how does this synthesis happen, actually? Well, if you think back for beta oxidation, that pathway was all about adding oxygen and taking electrons. It's beta oxidation, adding an oxygen in the beta position and taking electrons. Fatty acid synthesis is about removing oxygen in the form of water and adding electrons, or reducing the carbon. So beta oxidation, oxidizing the carbon. Fatty acid synthesis is reducing the carbon. Again, everything is about electrons. Metabolism is about electrons. Basically what the cell is doing is storing electrons in the hydrocarbon chain of the fatty acid, which is the best way to store these electrons in our body, and later on we can use it when we need it. So during beta oxidation, we had oxidation, then hydration, and then another oxidation reaction. In biosynthesis, we have a reduction followed by dehydration, and then followed by another reduction. So you can see, in principle, beta oxidation and biosynthesis of fatty acids is pretty much the opposite. But like I said, the machinery and the location is different. So what happens? Fatty acids biosynthesis basically happens, again, similar to beta oxidation, in groups of two. Carbons are added in groups of two, and the carbon chain grows in groups of two. In this, for each addition, for each cycle, 2 molecules of NADPH are used and a molecule of water is released. Think about beta oxidation. 1 molecule of FADH and 1 molecule of NADH were produced, and 1 molecule of water was used. This is the opposite. 2 molecules of NADPH will be used, and 1 molecule of water will be released. So again, I'm highlighting, in principle, they are opposite, but the machinery is different. So what is the enzyme that is used for catalyzing this reaction? Fatty acid synthase 1 is a single multi-domain polypeptide that catalyzes 7 reactions. 1 polypeptide, multiple domains. Why would you want to have multiple domains? Because you're catalyzing multiple reactions. And if you remember, a domain is a section or an area of a polypeptide that has a dedicated function to it. And a lot of times they're independent. In this case, they're not completely independent, but they still have these domains that have a dedicated function to it. It acts as a homodimer. In other words, 2 of these copies are bound together. So let's look what happens in brief terms. The first reaction is the fusion of acetyl-CoA with malonyl-CoA, and carbon dioxide is released. Then we have, again, a reduction. Electron pair is invested. Then we have dehydration, where a water molecule is removed from the beta place, beta carbon. And then we have another reduction to help, basically saturate that double bond. So we could probably easily call this process beta reduction, because that's exactly what happens in principle. This is the alpha carbon, and this is the beta carbon. What did we do? We reduced it. So in a way, we can call this a beta reduction, even though that's not an official title. I'm just saying it to make a point. All right, so fatty acid synthesis would always lead to a single product. Whenever we get a long enough chain, that chain is released. The most common fatty acids produced in our bodies is palmitic acid, which has 16 carbons. So metabolically speaking, what is the difference between the first 2, which end up being the last 2, or the 2 carbons at the tail of the fatty acid, and then the rest of the carbons of a fatty acid chain? When you look what happens, the very first reaction, we have an acetyl-CoA that has been bound by the enzyme, and then that acetyl-CoA will be added to malonyl-CoA. So if you watch the yellow highlighted section of the molecule, This is the only part, if you keep looking further and further, that's the only part that for the addition of it, directly acetyl-CoA was used. For all others, malonyl-CoA was used. Someone would say, yeah, but we used acetyl-CoA to generate malonyl-CoA. That is true. So in essence, we do add one acetyl-CoA at a time. But before we add it, what needs to happen to it? The acetyl-CoA was activated by carboxylation. Because of this carboxylation, then the fusion process can happen. That's just one way to do it. But each of the next acetyl-CoAs have to be converted to malonyl-CoA by carboxylation. Okay, so again, the tail, the first two added, or the tail, the last two on the tail of the fatty acid molecules come directly from acetyl-CoA. All the rest come from malonyl-CoA. Again, fatty acid synthase 1 catalyzes all of the steps in the production of a fatty acid, and that is all because of these multiple domains. And a critical role plays the acyl carrier protein, the ACP protein, which is quite flexible, and it can actually move the compound or the growing fatty acid molecule from domain to domain for a different reaction to occur. So now let's start with the detailed reactions. So what needs to happen first is both the acetyl-CoA and the malonyl-CoA has to be bound to the enzyme. The binding of acetyl-CoA, I mean, it's activated, but it's not bound yet. But by binding it in a way, we retain that activated state of acetyl-CoA. It's bound to the Ks subunit. The malonyl-CoA is then bound to the ACP carrier protein. And then they are placed, or they come into very close proximity. And when this negative charge on the carboxyl group collapses, then this electron density to the that is part of the methylene carbon here will attack the carbonyl group of acetyl-CoA, and the synthesis will be complete, because then this bond will be severed with the Ks, and then these two carbons will be added to each other. So that's the first step. And it's called activation of acetyl-CoA and malonyl-CoA because, in a sense, the binding of these two compounds to the enzyme is an activation. All right, so let's look at the rest of the steps. But before we move forward, I wanted to ask you these two couple of questions. So what is then the role of carbon dioxide? You saw in this first step, carbon dioxide was released. Once the methylene carbon attacks the carbon of acetyl-CoA, carbon dioxide is released. So what is the role of carbon dioxide? It was added, and actually its addition was paid by ATP. So what does that mean? That we invested energy and we activated the methyl group of acetyl-CoA, making it possible for the condensation to occur then later on between malonyl-CoA and acetyl-CoA. So the only reason why carbon dioxide was added so that it will be released. But before it's released, it activated the molecule so that it could perform work later on. All right, so the two are now added. The two acetyl groups are now added into one. The next reaction is, though, we have to remove now this oxygen. So how are we going to do that? First, we're going to reduce the molecule. So we're going to reduce the oxygen on the beta position to a hydroxyl group. Then we will dehydrate the molecule. The molecule is dehydrated. Water is released. but then what's left is a double bond between the alpha and the beta carbons. To saturate that double bond, another pair of electrons will be added to the carbons coming from NADPH. The first one also came from NADPH. And then we have a single bond in a fully saturated hydrocarbon chain. Now that molecule now is ready for another cycle. Then it gets, the acyl group now is transferred for the ACP to the KS domain. Here it is attached to the KS domain. And the ACP then picks up another molecule of malonyl-CoA, and the process continues. So, I think I missed one question here. Before this transfer of the molecule to the KS subunit, I was asking, what is the purpose of the previous 3 steps? So, and the answer to that is, it is to get rid of the oxygen. It's very simple. During beta oxidation, all the processes, all the 3 steps, the oxidation, the hydration, the oxidation again, their purpose was to take the electrons away, but also to add that oxygen. The purpose for this, the purpose of this process is to remove that oxygen in the form of water. I think I've missed also answering this question. Where did carbon dioxide come from? I probably answered it, but I can't remember whether I actually answered it. So I'm just gonna go back a little bit, and I'm just gonna tell you that if you remember, this was the carbon dioxide that was added to the acetyl group. This was the carbon dioxide that was added to the acetate to activate it. And again, the only purpose is so that it would activate that middle methylene group. So when the carbon dioxide is released, the remaining electron density will be used to attack the electrophilic carbonyl group and then form a covalent bond with it, shown right here. Okay, so the last question is, which carbon will serve as an electrophile during the next condensation? Now imagine this situation, but instead of a acetyl-CoA, you have this group now added to the KS. Which carbon do you think will be attacked? Well, the easiest question is, when you look at this molecule, which of these carbons is the most electrophilic? Well, obviously this one. So the reaction will always happen to the very first carbon that is bound to the KS subunit, because that's where the carbonyl group is. And you can see how each addition happens to the most inner carbon, and the next, and the next, and the next, until you end up with a carboxyl group at the end. This molecule eventually will be released as a fatty acid. In this case, in our bodies, the most common of those is palmitic acid. It's released from the ACP, the final step, is released by hydrolytic activity of thioesterase. One molecule of water will be used here to replace basically this sulfur, and the ending of the fatty acid now will have a carboxyl group as its end. I'm talking about the final process, not the cycling processes, but the final process. Alright, so how many cycles are required for a palmitate synthesis? Now, in order to answer this question, you need to know how many carbons of palmitic acid has. It has 16 carbons. So that means that you're going to need 8 acetyl-CoAs in order to make a palmitic acid. How many cycles, though, you need to go through? The answer is 7. And the reason for that is, it's not 8. The reason for that is because in the first cycle, our first product is we form 4 carbon group in the first cycle. And then the last one, basically release the molecule. But because of this, the fact that the first cycle unites 2 acyl-CoAs at once, and all the other cycles just adds 2 carbons at a time, that's why we need 7 cycles. Anyway, you can do it slowly, and you can prove to yourself whether I'm right or wrong. So for palmitic acid, 7 cycles. Or you can think of it as like 16 divided by 2, 8 minus 1. Same the way we used that formula previously when we were talking about beta oxidation. Okay, so the net reactions, the net substrates used, acyl-CoA and malonyl-CoA. We need 1 acyl-CoA and 7 malonyl-CoAs. Total 16 carbons. How many carbon dioxides? 7, because we have to activate 7 acyl-CoAs to convert them to malonyl-CoAs. ATPs, 7, because we have to do 7 activations. And NADPH, 14s, because we have 2 reduction reactions, both using NADH. Protons, 14. Products, how many palmitic acid molecules? 1. How many carbon dioxides? Of course, 7 will be released. ADP and PIs, 7 each. NADP plus 14. CoAs, 7. Water, 6. Why 6? Because even though we have 7 cycles, so we're going to get 7 waters, at the end 1 water will be used to release the final fatty acid from the ACP carrying protein. Okay, so interestingly though, I don't know if you've thought about it at this point, but again, fatty acid synthesis occurs in the cytosol. And fatty acid synthesis requires acyl-CoA. But acyl-CoA is produced in the mitochondria. So there has to be a way for that acyl-CoA to be taken out of the mitochondria into the cytosol if we're going to use it for fatty acid synthesis. Because the acyl-CoA that stays in the mitochondria is used for 2 things: making energy or making ketone bodies. So what is the source first of acyl-CoA? Let's see if you can think of that. What produces acyl-CoA? The precursor for acyl-CoA is pyruvate, if it comes from glucose. Pyruvate dehydrogenase converts pyruvate to acyl-CoA. What else? What other sources can we use to make acyl-CoA? Well, amino acids, the carbon skeleton of amino acids. We can use that to make acyl-CoA. Can we use fat to make acyl-CoA? The tone of my voice probably suggests that no, we cannot. But when we break fatty acids, we make acyl-CoA, so why can't we use it? Well, remember, those 2 processes are not active at the same time. If you have beta oxidation turned on, then fatty acid synthesis is turned off, and vice versa. So you cannot have fatty acid synthesis going active and at the same time have beta oxidation going active. They basically will neutralize each other. So all the carbon dioxide, also you have to think about when is one process activated, when is the other activated. When you make fatty acids, that means that you have excess of energy, and that energy has to be stored somehow. And why do we break fatty acids? We only break fatty acids if the body needs to use them for energy. If we need energy, in other words, there's no excess, there's a need, and that's when we break them. So these 2 processes almost never overlap. There might be just a little bit of overlap, but they usually are never active at the same time. Just like glycolysis and gluconeogenesis are not active at the same time in the same cell. The same thing is for fatty acid synthesis and beta oxidation. Alright, so how is then acetyl-CoA transferred from the matrix to the cytosol. This is a process that is ATP dependent. So familiar reactions, actually all of these reactions are familiar to you, will be familiar to you. Acetyl-CoA is bound to oxaloacetate by a familiar enzyme, citrate synthase, to form citrate. Citrate is exported outside. Once in the cytosol, citrate by the enzyme citrate lyase, that's the only enzyme you probably not, well, there are a few actually enzymes that you're not familiar, but citrate lyase converts citrate back to oxaloacetate in acetyl-CoA, but ATP is used for this process. The oxaloacetate is then converted to malate with the enzyme malate dehydrogenase. This is actually a fairly favorable reaction. If you remember, the reverse reaction that is used in the TCA cycle is not very favorable. Oxaloacetate is only a fraction of malate will be converted if left to reach equilibrium to oxaloacetate. So the reverse reaction is actually more favorable. So malate dehydrogenase easily converts oxaloacetate to malate. In this process, NADH is used because oxaloacetate will be reduced to malate. And now here is where the process actually, or the pathways here, divide. Malate could be transported inside the mitochondria and then be converted to oxaloacetate, again with the help of malate dehydrogenase, and then NADH is produced. Or malate can be converted to pyruvate with the help of an enzyme called malic enzyme. And in this process, malate is oxidized to pyruvate, and a pair of electrons are loaded on NADP+, forming NADPH, and of course carbon dioxide. Pyruvate is then transported back into the mitochondria. Pyruvate dehydrogenase converts pyruvate to oxaloacetate. Pyruvate carboxylase, I misspoke. Pyruvate carboxylase converts pyruvate to oxaloacetate. This, if you remember, this is the first reaction of gluconeogenesis, where we carboxylate pyruvate and one molecule of ATP is used in order to convert it to oxaloacetate, then oxaloacetate is converted to PEP, etc., etc. So this is a familiar enzyme. And then oxaloacetate is regenerated and it's ready to accept another acetyl-CoA. I mean, this first reaction is basically the first reaction of the TCA cycle. And when would citrate actually be taken out? Well, if you think about it, it will be taken out when you have plenty of it. If you have too much, if you have plenty of energy and citrate is no longer needed, then citrate, it'll be easier to be transported. And when it gets transported out, it gets immediately converted back to oxaloacetate, acetyl-CoA now that has been used from inside the cell, let's say from glycolysis, it gets transported into the cytosol. And then how will that acetyl-CoA be used now in the cytosol? To make fatty acids. So you can see a very easy connection between glycolysis and fatty acid synthesis. And these two processes occur at the same time. No problem. So again, you don't have to eat fats. Even if you eat fats, they will not be degraded and then repackaged as a fatty acids. The fats, and I still remember my pathophysiology professor that says that the best way to gain weight is to eat bread and butter. Why? Because the bread supplies the carbohydrates, which produce plenty of energy for the body, and the butter just gets packed in deposits, fat deposits. Why? Because you're not going to use the fat. I mean, you use some of the fat, obviously, for muscles and heart, but that's not a lot. It depends how much you consume. And so if you eat a lot of carbohydrates and fats, the easiest way to gain weight. The modern bread and butter is ice cream, because ice cream has great amounts of sugar and substantial amounts of fat. So basically, you pack those two together, perfect recipe for gaining weight. So think about when you eat your ice cream next time. I know that's not going to change your mind. It doesn't change my mind either. I'm just saying that that's the best way to gain weight. All right, so how many ATPs are then needed for the addition of each two carbon groups to the growing fatty acid molecule? Well, think back. To convert acetyl-CoA right here to malonyl-CoA, you will use 1 molecule of ATP. But to export a molecule of acetyl-CoA from the mitochondria to the cytosol, you're going to need 2 molecules of ATP. Now someone might say, well, what if malate follows this route and not the route of pyruvate? That's true. It could follow that route, but it won't. Majority of it will follow the route of pyruvate. And now this brings us to the next question, the significance of that. Well, look at the product of this when malate becomes pyruvate and then pyruvate gets transported versus malate being transported directly. The product is NADPH, which is needed where? In fatty acid synthesis. So that's why malate will prefer, or the cell prefers to go the route through pyruvate, not just directly to put malate in. Now, if malate gets put directly in, what's the benefit of that pathway? We're transferring NADH inside the cytosol, inside the mitochondrial matrix, and that NADH now can be used for optimal amount of energy production. But in our case, when we make fatty acids, we already have plenty of ATP. Why would it matter to put just one more electron pair inside? We already have plenty. That's why we're making fat, because we don't need more energy. Under these conditions, under these circumstances, the malate will prefer to become pyruvate to generate NADPH that will be used in the fatty acid production. Okay, now to get to the NADPH part, so obviously one side of the story, acetyl-CoA, we just transported, you can see it here, we've transported acetyl-CoA from the mitochondria to the cytosol. But now where does the NADPH comes from? You already know part of the story, and that is the pentose phosphate pathway, which is active during glycolysis, especially if we, or if the fatty acid synthesis is activated, which uses NADPH, and that helps now glucose 6-phosphate to enter the pentose phosphate pathway to generate more NADPH and basically get burned to carbon dioxide right there instead of entering to the TCA cycle, with the whole point of it being to generate NADPH and carbon dioxide. That pathway provides about 50% of the NADPH needed by fatty acid synthesis. The other 50% comes through the malic enzyme reaction from malate to pyruvate, this reaction. So you can see that this reaction is essential, is required in a way for optimal fatty acid synthesis to occur. So that's why malate primarily will go through the route of pyruvate instead of being directly transported into the mitochondria. Alright, so 50% pentose phosphate pathway, 50% the malic enzyme. In terms of regulation, obviously fatty acid synthesis gets activated when we have plenty of acetyl-CoA and plenty of energy. More specifically, citrate actually activates acetyl-CoA carboxylase in addition to the activation by insulin. If you remember when we've talked about the carnitine shuttle, we mentioned then that the ACC enzyme, acetyl-CoA carboxylase, is activated by insulin. And it converts acetyl-CoA to malonyl-CoA. Citrate also activates that enzyme. Why would citrate activate the first reaction of fatty acid synthesis? I mean, citrate is a molecule of the TCA cycle, which is the burning molecule. Well, again, it's quite logical. Think about it. If energy is needed, citrate will be converted immediately to isocitrate and it will be burned in the TCA cycle. When will we have higher concentration of citrate, higher in order to activate the acetyl-CoA carboxylase? Because usually that's the threshold. It has to, the concentration has to rise above certain level in order for it to bind to its binding site and then activate the corresponding molecule enzyme. That concentration of citrate will rise above that threshold when the cell is basically telling the mitochondria, oh, stop, no more, we have plenty of ATP, start storing. The TCA cycle will be inhibited, citrate concentration will rise, and then in a way you can think of it like it'll spill over through the mitochondrial membranes into the cytosol, where it basically will be like, okay, even though this reaction is favorable, citrate synthase, we're going to transport it out, we're going to dump the acetyl-CoA now back in the cytosol, and then that will be used for fatty acid synthesis, and we'll bring back oxaloacetate to malate pyruvate, and then get back to oxaloacetate. Oxaloacetate again will accept another molecule of acetyl-CoA if we have plenty of it, because of plenty of glucose in the system, and then we'll bring it out. So basically, again, what is happening? We're making acetyl-CoA from sugar, and we're packing it as fat. So you can see again the flawed idea that if you don't want to get fat, don't eat fat. That's actually quite confusing and wrong. I still remember in the late 90s, when the 80s and 90s were the hyperactivity. Well, people were quite focused on low fat and fat-free everything, even butter, I've mentioned before. And I would see them eat a lot of sugar, like candy bars and drink soda, but oh no, we cannot eat fat. Again, such a flawed idea that if you eat fat, you're going to get fat. Well, again, think about the keto diet, how it is. They tell you basically eat as much as you want fat and protein, just don't eat carbs, and you will lose weight. And indeed, that's what really happens. And it's all because of the absence of carbohydrates, which are the primary energy source. And here you can see it again. Acetyl-CoA is a product of carbohydrate degradation. And then that extra acetyl-CoA gets packed as fatty acids. So you don't have to have, and I already mentioned that even if you eat them, if you have carbohydrates, the fatty acids will not be touched. They will be repackaged into triglycerides and shipped out into the deposits. They will not be touched. They will not be degraded. You just package them, repackage, and ship them out. So for that reason, citrate is an activator. If the citrate goes up, you want to stimulate the acetyl-CoA carboxylase in order to use acetyl-CoA so that transport out in the mitochondria will be activated and more of extra acetyl-CoA will be taken out. Also, the increased malonyl-CoA would inhibit beta oxidation since there is no need for fatty acid degradation. So that's just another added benefit of that. Okay. Now let's look at it a little bit from a larger perspective. So I told you that the palmitic acid is the main product or the most common product of fatty acid synthesis in our bodies and in animals. Now, from there then, our cells can generate longer amino acids by extending the 16 carbon palmitic acid to steric or even longer, or that palmitate now can, or steric acid can be desaturated, and then we can generate the palmitoleic acid if it's from palmitic acid, or oleic acid if it's from steric acid. So we can generate monounsaturated, we can generate longer and monounsaturated fatty acids, but our bodies and animal bodies cannot synthesize polyunsaturated fatty acids from this precursor, which is palmitate or steric acid or oleic acid or palmitoleic acid. I mean, these are the most common fatty acids from animal products. Steric acid, coming from beef tallow, steer, you know what that is basically. Oleic acid, in oils, in pork or whatever. But our bodies cannot use these amino acids to make polyunsaturated fatty acids such as the linoleic and alpha linolenic fatty acids, which are essential. We cannot make them. We cannot further desaturate what our bodies can make in order for us to use now the essential fatty acids to make other bioactive molecules. These linoleic and alpha linolenic acid, they have to be consumed with plant food or other food, whatever it is that contains them. The plant cells can synthesize polyunsaturated fatty acids, but not animal cells. Now, once we consume the essential fatty acids, then we can further desaturate them and elongate it, and we get other bioactive molecules, a major one of which is the arachidonic acid. Now, arachidonic acid is a special molecule because it is a precursor for a large group of bioactive molecules such as prostaglandins, thromboxanes, and leukotrienes. These are molecules that are involved in inflammation, in immunity, in pain signaling, and in all the different variations of inflammation and pain regulation and temperature regulation, etc., etc. You can see with the help of the enzyme called cyclooxygenase, or COX, in this case is COX-1, even though it's not shown here, you can convert arachidonic acid to prostaglandin G2, then to H2 or other prostaglandins or thromboxanes. I mean, they're, like I said, they're involved in the immune response, in biosignaling, in platelets activation, in blood clotting. I mean, you name it, tons of processes that are regulated by leukotrienes, thromboxanes, and prostaglandins. Now, for that reason, this enzyme, the COX-1 enzyme, has been a target for the pharmaceutical industry. As you can see here, aspirin and ibuprofen and another molecule, as a matter of fact, can inhibit it. Naproxen is the other molecule that I forgot for a second. So, like I said, it's a targeted enzyme. I mean, who of all of us has not taken aspirin, or I guess in the United States, Advil, which is ibuprofen, or Aleve, which is naproxen? Who of you, or all of us, has not used those? I mean, we use them quite often. Every time you have a headache, okay, we'll pop one. Every time you have a fever, ever since, a person is born, basically, you give him. Well, actually, you don't give aspirin or Advil or Aleve or none of these called non-steroidal anti-inflammatory drugs. You don't give these to pregnant women and children that are young, because the fetus in pregnant women and young children specifically, they cannot process aspirin. Their livers are not quite yet developed to be able to process the salicylic acid, and and they can develop a toxic reaction. So that's why we don't really give aspirin to little children. Ibuprofen, Motrin is the company name, the brand name. Ibuprofen can be given for young children, but usually people prefer to use Tylenol, which is acetaminophen, acetaminophen is not a non-steroidal anti-inflammatory drug, so that's why it's not added here. However, the problem with these medications is that they have a very bad side effect. But that side effect only happens if you use the non-steroidal anti-inflammatory drugs or the COX-1 inhibitors for a prolonged amount of time in high doses. And that usually happens with people who have rheumatoid arthritis. And people, those people, they take these drugs in large quantities and for a prolonged amount of time, and they develop stomach complications, gastritis, ulcers, and sometimes they get complicated. They might perforate or bleed, and you can lose your life because of that. So for that reason, the pharmaceutical industry and doctors in general have been looking for a better way to inhibit prostaglandin production. Someone might say, well, what does prostaglandins have to do with stomach mucosa? Well, like I said, these are molecules that are involved in a wide range of processes in the body. I mean, they're part of the inflammation signaling. That's why you take the inhibitors to suppress the inflammation. You take them for headache because they're also involved in pain signaling. You take them when you have fever because they're also involved in the signaling during inflammation and the temperature control. So by inhibiting them, you actually drop the temperature. So that's why you take them for. That's why you inhibit this pathway for so many different things. I mean, some people even take low doses of aspirin because it's believed that the COX-1 is somehow involved in the cancer pathways in the colon cancer. There's studies that show that taking low doses of aspirin after you're 40 or 45 and for the rest of your life, it actually decreases the onset of colon cancer. Now, I have no idea whether this is really true, but there is a suggestion that that's what really happens. And so these are drugs that are used for many things. But again, if you take high doses for a prolonged amount of time, no matter how you take them, right? We joke sometimes that whether you take them from your mouth or the other end, the effect is still the same on the stomach. The prostaglandins are involved in the proper production of mucus that protects the stomach from its own acid and enzymes. But when that production is now decreased because you're suppressing prostaglandins, the protective layer in the stomach is now worn out, and the acid and the enzymes now have a direct access to the epithelium of the stomach and anything underneath, and so they start degrading it, and that's how ulcers are generated. So for that reason, the search for a different inhibitor has been so, valuable. And that inhibitor was actually found, and it was first discovered by a professor at BYU, actually, cyclooxygenase 2. And that enzyme was believed, by inhibiting that enzyme, we would achieve a very specific, well, I shouldn't say very specific, because what I'm about to say, but we can achieve a relief from inflammation without affecting the stomach. And that was a revolution. Everybody was so excited because now you can control inflammation from rheumatoid arthritis without affecting the stomach. Well, I always say that no matter what you take in the body, there is a desired effect and there is an undesired effect. I have not so far come across of a compound that only has a desired effect without side effects. And these medications, these three actually, Vioxx, Bextra, and Celebrex, they also had their own set of side effects. And in the case of Vioxx and Bextra, these side effects were actually in some cases lethal, because they will affect the valve structure of the heart, and people that will take them, some even died suddenly. And as a matter of fact, Bextra was taken out very quickly off the market. Vioxx stayed for a little bit on the market, but then it was taken out too, and there was a lawsuit with Merck that makes Vioxx, I believe it was Merck, and they had to settle it for $2 billion. So now both of these drugs, Vioxx and Bextra, are off the market. Celebrex, however, is still on the market, but in a black box, which basically tells the person that takes it, take it at your own risk, you've been warned. But there's still people that take it, they're just more controlled, in a more controlled environment, and they're monitored. And these are probably people who cannot basically use the non-steroidal anti-inflammatory drugs, and they have to basically take Celebrex. Interestingly, it's an interesting story. At some point, I don't even remember what was the reason. I think I had a back problem or something. I was at BYU during my graduate school at that time, and I was meeting with one of the doctors at the Student Health Center, and he gave me samples of Celebrex, like a handful. I think I had like a 9 or 10 samples, and I had told myself, oh, I'm not taking this, because I have this unwritten rule for me: if you can, don't take a brand new drug. Now, if that's the only option for you and there's nothing else, like for example this vaccine now, I can understand, that's just basically nothing else on the market. But if you can choose something else that is more established and can do a similar job compared to a brand new drug, don't risk it. And in this case, I took the Celebrex, I kept it, but I never used it. Then I ended up throwing it away. And later on, I figured out that, Celebrex was placed in the black box. This was right around the time when Vioxx lawsuit was happening. I wish now I kept it so that, I can have a memory of that. But anyway, so that's the story with the COX-2 specific inhibitors. It's just another isozyme, isoform of COX-1 that had a more specialized effect on the inflammation without the stomach lining complications, but it turned out that it has other complications that are, just as lethal in a much shorter time, actually, than the COX-1 inhibitors. All right, well, this was a little longer. It's now over an hour. I don't think you've ever had a lecture over an hour in this semester. But most of the things that took that time were, talking about these medications, and I allowed myself to talk about more extensively about these because in class I can't. I just mentioned them, actually, very briefly, because it's a 50-minute class. But since it's online now, and I have a lot of time on credit, I didn't worry myself too much about it. So, but yeah, I'm sorry that it took me a little more time. Hopefully it was interesting, though. And everything was clear. Again, as always, if you have any questions, let me know and I'll be happy to help.